A warm welcome and very good evening to all dear participants. Resource person of today's session, Dr. Jyoti Batra ma'am, and chairperson of today's session, Dr. Uma Bharadwaj ma'am. Now, I would like to introduce today's resource person, Dr. Jyoti Batra ma'am. Ma'am is presently Dean Research and has been associated as Professor of Biochemistry with Santosh deemed to be University, Ghaziabad, for more than 23 years. Here, she has delivered lectures, contributed in course enrichment, and supervised many doctoral researches. Dr. Batra has filed uh, patents and copyrights and have published a rich number of research papers in Scopus, PubMed, Web of Science, and other index journals. She headed Central Research Facility of the university, headed the Bioethics Unit of the university, and this research policy and doctoral research procedures started and heading incubation center in the university and funding support up to 1.5 lakh for students team innovative idea by new generation IEDC for developing prototype and incubator supported by DST. Ma'am supported and monitored seed money allocation, distribution, and utilization. Started maintained student research cell, motivated and maintained rich numbers of publication in Scopus, PubMed, and Web of Science, and maintained high H index of Scopus, 20 plus Scopus index. During her tenure as a research dean, she has conducted multiple workshops, measure out of the listed like Section 8 company types and establishment process and orientation on government schemes, orientation program, and insight into criteria 3 now, orientation program, and overview of Scopus and PubMed, and overview of PhD program, and step and processing of intramural grants. Like there are many other events organized by the Santosh University. Ma'am has awarded various awards like she has awarded exemplary research award in 2021 International Association of Research Scholar. In 2021 award as Innovative Excellence Award by Innovation Society, the International Certificate on Principle of Ethics and Human Rights of Award, UNESCO Universal Declaration of Bioethics and Human Rights in Medical and Health Science Education by UNESCO, Chair Bioethics, FIFA Department of Education in 31st October 2020, and awarded as International Association of Research Scholars is an international association for researchers from diversified fields of studies dedicated to play influential role in global promotion of research activities in diversified areas of studies. IARS recognized the inspiring contribution of Dr. Jyoti Batra's thought its New Delhi chapter towards support and spread of researchers in biochemistry and honor her efforts with the prestigious IARS SPGN Research Scholar of the Year 2020. Dr. Batra receives a letter of appreciation along with Harmony Research Grant of $50 USD on 15 July 2020. And Dr. Jyoti Batra ma'am received the INC Research Excellence Award 2020 by the Institute of Scholars INSC that is an emerging technical professional organization established in the year 2014. Ma'am is awarded by American Association of Higher Education, one of the oldest existing academic NGO, has recognized efforts of Dr. Jyoti Batra ma'am towards uplifting education and research standard through continuous support to students, scholars, groups, and organizations for highlighting the latest research issues and the exploration of optimum solution. Dr. Batra ma'am receives letter of appreciation from AHEA. There are various number of awards received by Dr. Batra ma'am. Now,
You can cut it short.
without taking too much time, I have no words to explain these all things. Without taking too much time, I request to ma'am to please lead today's session on the topic biochemistry. Please, ma'am.
First of all, thank you so much to Institute of Applied Statistics for giving me this opportunity uh, so that I could at least come to this forum and talk about, something different. Believe me, I was asking Dr. Shubham, like, what I am going to do in Ayurveda. I have never been a Ayurveda person, and I don't know what to say. So he encouraged me, so I am here. Thank you, Dr. Shubham, for that. And I want to actually acknowledge the presence of our chairperson also, Dr. Uma from NIU. Um, Happy to be there sharing the same dais with you, ma'am. So the topic which has been given to me is biochemistry approach to Ayurveda. I'll try to do little justice to whatever has been given to me. It's up to you, and you are open to the questions. Okay. So, as we all know since beginning, that Ayurveda talks about only three things: Pitta, Vata, and Kapha. I have heard about these things a lot in my childhood and growing up time, but I can say, like, being from a medical background and a family of medical professionals, it was absolutely, um, only dadi nani thing, that if you are having some kind of a cough, okay, you take some ginger with honey and all. But there was no, that belief into that. But nowadays, I mean, there is a lot of effort is going to improve and uplift the Ayush part of the Ayush means Ayurveda part of the research and coordinating it with the medical finding. However, a lot has to be done in this field. So I'm trying to put forward the communication between Ayurveda and medical research. So if we say about the etiological factors responsible for different diseases and their development has been described in detail in the Ayurvedic system of the medicine. Health here has two classified forms. One is physical and another is psychic. We don't only talk about mental and body health. The four basic causes of the onset of different diseases is genetic, psychosomatic, environmental factors, abnormal activity of sensory organs. It's as similar as we talk about medical allopathy science of clinicians. For the management of those factors which involve the imbalance of Vata disease, a particular way of life and a special group of medicines are recommended, which are called as Rasayana drugs. The actual meaning of Rasayana is to follow those things which prevent aging and keep an individual healthy. I mean, prevent aging is something which everyone wants in present life. And when we talk about preventing aging, you must have seen a lot of commercials showing that if you use this kind of beauty products or if you will eat this kind of food, you will at least you have to age. Everyone has to age, but there will be a prevention of the aging part. Why and how it happens, at least in Ayurveda, it was not clear. But it is there, that is for sure. They have few theories, like the increase in Vata is due to intake of unwholesome diet, excessive exercise, excess stress, poor nutrition, and that leads to early aging in the metabolic diseases like my diabetes, arthritis, Alzheimer disease, loss of memory, etc. And Vata is also said to be increased. These the levels of some rasayanas or chemicals in your blood or inside your cell than the normal levels, right? It includes the medicine, the diet, and also the lifestyle. Interestingly, aggravated vata could be checked by taking special diet, stress-free life, holy thoughts, etc. Approach of treatment is always to change lifestyle and use of some concocted medicines which has been given by Vaidyas not only cures the disease but also keeps a man healthy for a longer life. Nobody knows how. The basics were not connected. Interestingly, this concept of Ayurveda resembles with free radical theory of disease and aging. Free radical. What is free radical? As it is known that along with the evolution of living organism, the involvement of oxygen and iron in the life processes became more significant. When we talk about free radical, what is free radical? Free radical is those radical which have an extra pair of electron into them. And that is very, very dangerous to your health of the body. For example, we can say like when we clinically talk about cardiovascular disease, we say that there is good fat and bad fat. So if bad fat deposits in the veins or arteries, what happens? It will block them. Once it is blocked, there will be a heart attack. But what is that thing which is making these arteries narrow? We call that edema. Now, from where this edema is coming? So, when we talk about this edema is actually bad cholesterol or bad fat which has been oxidized by these free radicals. Now, from where these free radicals are coming? These are coming by the excess activity of electron transport chain, free iron if it is in excess, or if there is hyperoxia, pollution, radiation, stress, excessive exercise. There are so many factors which can increase the production of these free radicals. And these free radicals are going to make a havoc in the body. All toxic substances are going to be produced which your body is not capable of detoxifying. We have our liver which is a very good machinery to take out all the toxins from your body. It has some reactions specific for taking these toxins out of your body, but there is so much excess of free radical that the number of toxins produced are much more than the capacity of the body to take them out. So that is why we call that free radicals are produced in a very high quantity because of these reasons, and they can cause disease and aging. Now, some of these end products of this process get eliminated from the body by the action of cellular proteases. As I told you, there are two phases of detoxification reactions in the liver, and they completely remove whatever toxins are produced as a end product of metabolites. However, some of them become unfit for the natural constitutive proteases and lysosomal enzymes. Everyone who has a little knowledge of the cell structure, they know lysosomes, which destruct all the toxic part of the um, cells. Whatever toxins are produced, they break it and then they excrete it out of the cells. Now, this is not happening because there is excessive production of free radicals. So what happens? These get accumulated, and because of their inability to enter to the normal metabolic pathway or elimination, they now start entering into different systems. And once they enter into these systems, they give this extra electron and start producing superoxide radicals. Because of these superoxide radicals, these toxins are now not healthy for your body. They will start depositing into the lines of your um, arteries or in the brain also. So many places you will find that these toxins are going to affect the normal metabolic reactions. As per recent scientific information, there are basically three mechanisms which are involved in atherosclerosis. One is lipid infiltration, second is cellular invasion, and cell proliferation, which is similar to maida deposition and coverage of channels. This is how we connect it to the Ayurveda. कि क्या हो रहा है आपके बॉडी में, लाइक कुछ मैदा डिपॉजिट हो रहा है. व्हाट इज दिस मैदा? दिस इज एथिमा, which is narrowing your arteries, and because it is narrowing, the pressure of the blood is high blood pressure is reported, and this high blood pressure may convert into the heart attack when a small part of this edema will break and block any part of the thin artery. One of the hypotheses is the involvement of free radicals and nitric oxide. Nitric oxide in Ayurveda we call it high vata, which may be due to high concentration of advanced glycolization end products. So what happens? Glycolization is a oxidation of carbohydrates. It again produces toxic end products. So these can also end up into the free radical production. Yes. The oxidized LDL in blood stimulates the monocyte binding. I'm not going into these details. As I've told you, LDL, that is low density lipoprotein, which is a bad cholesterol, for your body, which this is a bad cholesterol, but when this is oxidized by free radicals, it becomes super bad, and it produces chemotactic factors, which is in Ayurveda we call it high pitta, of high enzymatic activity for addition of monocytes to the endothelial cells. Endothelial cells are the membranes of these your arteries. Similarly, when we talk about the lifestyle and diet, then at biochemical level, hurry could be correlated with the high activity of mitochondria, producing high amount of superoxides as the byproduct of electron transport chain. What is electron transport chain? It is a process of production of ATPs. Why do you require ATPs? Because ATPs are those molecules which will break down to give energy for each and every function of your body. Even if you want to, release your you want to shift your hand from one place to another, you need some ATPs to do it. I'm utilizing my ATPs, so ETC has to function for each and everyone. But when you are stressed, what happened? You are releasing some hormone which will activate your mitochondria to produce more ATPs, more energy you need. And when this is in excess, what will happen? As a byproduct, a lot of superoxides will be produced as a byproduct of electron transport chain that can affect your health of the body. Then worry, that is stress. It is related to high release of catecholamines. Catecholamines, everyone knows, these are the stress hormones, which on reaction with free iron in the body produce more hydroxyl radicals. Again, hydroxyl radicals are what? These are free radicals which can produce more and more toxins in a multiple way. Curry means food. This is indicating the excess intake of fat, which is the best substrate for lipid peroxidation because of more unsaturated bonds. Here a little bit difference is there in the basic theory, because in medical sciences earlier we used to say it is excess intake of fat, but now it has been replaced by excess intake of carbohydrates. In excess intake of carbohydrates, that converts into the fat as a storage. Triacylglycerol in your fat body deposits in adipose tissues, which is the substrate for lipid peroxidation, and By lipid peroxidation, anything will be oxidized when you have a double triple bonds. Okay, electron changeover will be there because your oxygen radical high oxidant will have always a extra pair of electron which will be utilized for this purpose. This is a little, I have taken it from some Google diagram which says in Ayurveda how it happens. So I don't have much idea about it, but ahar means food. When it is digested and absorbed, you get a lot of bile, and this energy goes to your rat, means your blood, your bones, your cells, etc., etc. And when there is excessive of lipid or fats, what will have, you will have lots of maida. Maida means, which we have discussed earlier also, this will be the ethema in the clinical language. Now, what is etiology of atherosclerosis? If we go by the Ayurvedic view, the mechanism underlines the modulation of various signaling molecules leading to activation of nuclear factor kappa B, which is a transcriptional factor. I know this is a little bit jargon here, but transcriptional factor is those factors which actually help to create from your DNA the RNA and the proteins. As you know, proteins are, one function of the protein is to help as a transporter and a regulator. So here this is involved in the regulation of immunity. There is two types of immunity, I think everyone knows. One is innate, when another is acquired. So this nuclear factor kappa B is involved in the regulation of innate immunity, which affects the expression of genes. And these are responsible for inflammatory factors and chemotactic agents, which are VCAM1, LM1, and intercellular adhesion molecules. These are all clinical jargon, but in a wholesome term, we can say that these can be compared to high kapha, pitta kapha. So kapha, this is all kapha which is being produced, and when you explain this kapha, it is this nuclear factor kappa B, which actually affects your immunity. So basically, when the immunity is compromised, you have lots of kapha. Then, if we talk about brain, Alzheimer's disease, where person actually starts forgetting things, the three possible causes: free radical, amyloid, body deposition. These are some kind of structures which are formed inside the cell, and cell functioning is disturbed, and the calcium toxicity. If we talk about the molecular level, there is activation of, again, kappa B cells that will enhance the expression of one specific enzyme of electron transport chain, and ultimately the induction of inflammation in the brain tissue. Inflammation means there is, like, what is inflammation? If you have आप गिरे हो कहीं से और आपको थोड़ी सी चोट लग के थोड़ा सा वो रेडिश सा हो जाता है ना, that area, that is called inflammation. That means your body is trying to protect yourself against the bacteria entry into your body. So it starts inflaming itself so that it is protected. Now, beta amyloid bodies, which are amyloid body disposition, which you can see here, these are deposited inside the cell, and these are, what are these are transformed proteins. Again, how proteins are formed? These are formed from the genes. Like in earlier slide, we have just talked about genes. Like here it is expression of genes. This expression of genes is being compromised. When it is being compromised, what is happening? Your protein is being transformed, and what happens after that? There is a altered precursor protein is synthesized, which is accumulates and degenerates your neural cells, that results in amyloid body deposition, and this amyloid body deposition is the cause of Alzheimer's disease. Another factor is excess of calcium ion in the cytoplasm, which is the outcome of hyperactivity of glutamate receptor and also the calcium channels in the cell membrane. These also create a lot of, um, immunity problems in the cell structures. Glutamate is, what is glutamate? It's a kind of, neurotransmitter. And neurotransmitters in a very small amount can create a lot of disturbance in the body. This high cellular calcium triggers the production of free radicals. Again, calcium channels are disturbed. Now, calcium channels, what will happen? There will be a high calcium level in the cellular level. Once cells have high calcium level, they will cause the production of free radicals. Free radicals will cause lipid peroxidation. Lipid peroxidation, what is in general term, what do you understand by lipid peroxidation? You must have seen that घी का डब्बा जिसमें बहुत दिन तक पड़ा रहे और उसमें एक गंदी सी स्मेल आने लगती है. You don't, आप बोलते हो ना ये खराब हो गया इसको फेंक दो. So that is lipid peroxidation. That is what is happening inside your body, which is very very toxic. जब आप उसको सूंघ नहीं सकते, खा नहीं सकते, आप सोचो आपकी बॉडी के अंदर ये सब प्रोसेसेस हो रहे हैं बिकॉज आप स्ट्रेस ले रहे हो, आप एक्सपोज कर रहे हो अपने आप को पॉल्यूटेंट्स को, और यहाँ पे बहुत सारी चीजें जो मैंने शुरू में बताई, दैट कैन बी द रीजन फॉर दैट. Then high cellular calcium triggers this thing and the process of lipid peroxidation, and finally neuron damage. Again, we are talking about neuron damage, and neuron damage means Alzheimer's disease. Ayurveda, now what it says, it believes that mental disorders are the basic cause for increase in vata, that is prana, and the accumulation of ama. Ama means amla, means acid in the brain and the nervous tissue. They say now amla means acid. Acid means low pH. Low pH means high oxidants. So that is how it is all connected. It leads to the inflammation and degeneration of cells. It is a general principle for the development of Vata's disease, where it is said that the disturbed Vata affects the Agni and then produces acids. This is how this whole thing functions. Now, how to manage these diseases? Ayurveda describes the use of rasayan drugs. In this system of management, attention has been paid to this branch as it is anti-aging. Anti-aging, everyone wants that they should not get old very early. They should not age at all actually. It's not only recommends for the use of herbal medicine but also includes the code of conduct and lifestyle. Now, what is the use of herbal medicine and lifestyle is going to help you as anti-aging? Again, these herbal medicine contains some chemicals which are going to block the production of your superoxide radicals, that is oxidants, and these oxidants will be nullified so that they can not create such toxic effects in the body and anti-aging in nature. Interestingly, a range of herbs have been described for different age groups as rasayan therapy. It opens the channels of the body and leads to normal metabolism of ingested food. So what these herbs again do, they activate some enzymes, and these enzymes are now doing proper digestion and assimilation and absorption of the food content so that normal metabolism works. it brings vitality to all dhatus, that is body systems and their organs. These are the words taken from Ayurveda, but they in the clinics you use these kind of systems. You have number of body systems, you have digestive systems, respiratory system, sensory system. I mean, there are so many systems. It's further recommended for the regular intake of these medicines after the age of 25 to 30 as the diet supplement to prevent the aging process. So, what is basically is the aging process, how you are protecting this? These are actually reducing the number of oxidants that are produced due to this hectic lifestyle which we are leading today. You go out of the house and there is a lot of pollution there. What is this pollution is made up of? Oxidants, and they are going to enter into your body and creating superoxide radicals. They call it vata mediated disease in Ayurveda. On the similar pattern, nowadays the use of antioxidants has come to practice. It is both preventive as well as curative. Specifically, the medicines responsible for the management of free radical mediated diseases are called antioxidants. Can any one of you in the whole group tell me what are our natural antioxidants present in our body? Anyone? You can unmute yourself and you can tell me. What are natural antioxidants which are present in our body? Okay, I let me put it. Yes, glutathione. Okay, right. And some antioxidant vitamins. Do we have some antioxidant vitamins? Vitamin C, vitamin E, very good. Yes, yes, Dr. Bharti, vitamin C, vitamin E. Any other vitamin other than these two? Okay, okay, vitamin D. Yeah, that's this is what I was waiting for. Vitamin D being it's associated with hypervitaminosis. Yes, carotene, beta carotene, right, right, very good. So we have lots of these natural antioxidants. Even the uric acid is to some extent is a yeah, beta carotene. Right, right, right. Correct. So people know what are these antioxidants we have in our body. That's why they say somebody answered lycopene. Lycopene means you have to have colored, you know, fruits and vegetables. If the color is more into your food, you are having lots of antioxidants and it will be all anti-aging. So what these do, they inactivate reactive free radicals which are spontaneously being produced in the normal physiological processes, especially in electron transport chain. They also chelate the free metals and activate the endogenous antioxidants enzymes at molecular level. What are the antioxidant enzymes? We have a group of antioxidants enzymes in our body. Can anyone share some names with me? Somebody said glutathione. Oh, my voice is breaking. Hello, is my voice clear?
Yes, ma'am. Voice is clear. Dr. Rachna, I think there is network issue at your end. Please rejoin if there is a problem is still facing your
Yeah.
facing.
So I was just asking about we can have a discussion. What are the antioxidant enzymes our body is having just to protect ourselves? Somebody said glutathione. The name is very familiar with the enzyme also. Wow, very good, Rishabh. Glutathione, okay, okay. Peroxidase, yes, yes, yes. So we have these enzymes. These are the endogenous enzymes. And these enzymes at the molecular level function as antioxidants. They control either the production, yes, catalase, very good. They either control the production, superoxide dismutase, yes, I have a very learned audience here. Very good. So, what do they do? They take up these antioxidants and convert them into non-oxidant substances and take it out from the body. Here they, at the molecular level, if we talk, antioxidants attenuate the activation of kappa B and several other transcriptional factors, and gene expression is regulated. Activity of ion channels and different enzymes, especially the kinases, is also being regulated. And these, as you all of you know, must be knowing that kinases are the very important in the signaling cascade. If we talk about hormonal cascade or we talk about enzyme cascade, this is a very important role to play kinases. Interestingly, if we talk about herbal medicines, they have shown antioxidant property in the, for example, we talk about haldi. We talk about haldi, it has been worked upon a lot. So many publications are present nowadays which are talking about the antioxidant property of the, yes, karkuram. Yeah, yeah, yeah. So these are all the same. Only thing, we have to make a connection between these two. We have to, this amla, which is full of vitamin C, it is present in so many Ayurvedic medicines. Mulethi, it is present in so many Ayurvedic medicines. They all have full of antioxidants. It also acts as the signaling molecule by itself. In this way, it appears that most of the properties of free radicals are similar to that of Vata as described in the Ayurveda. So, a hypothesis could be made that the free radicals and related antioxidant enzymes and molecules could be treated as one of the objective parameters for the assessment of Vata and Ojas. Similarly, the other terms of Ayurveda could also be defined as some objective parameters. Like here we are talking about nervous system. If we talk about Vata, it is wind, nervous system, hormones, membrane-bound signal transduction, stimulus receptor interaction. Pitta is bile, thermogenesis, enzymes, phosphorylation processes, metabolic steps. Kapha is phlegm, thermotaxis and formation of preservative fluids, immune system, gene expression, defense system. So we can see that there is a connection between Ayurveda and the modern medicine. In Ayurvedic system of medicine, it is considered that a living system is made up of Panch Mahabhuta in the form of Vata, Pitta, Kapha at the physical level, and Sattva, Raja and Tama at the mental level. So we have started with physical and the mental level, if you remember the first slide. Again, we are coming to the same thing. This covers the psychosomatic constitution and commonly known as Tridosh theory. The imbalance in these body humors, that is blood, yellow and black bile and phlegm. Again, we are talking about Pitta and Kapha, is the basic cause of any type of disease manifestations. Till date, several objective parameters have been proposed to monitor the level of these basic humors, but none of them is complete. So a lot of studies are required to first identify this combination and coordination between Ayurveda and the, modern medicine, and then to prove it. So free radical theory may be considered as one of the objective parameters of the disease. To be more specific, Vata can be monitored in terms of membrane-bound signal transduction. What do you mean by membrane-bound signal transduction? Anybody? Membrane-bound signal transduction. When we talk about membrane bound signal transduction, what we are talking about here? You can give me in the chat box your answer. Pitta is as the process of phosphorylation and dephosphorylation of different proteins, signaling moieties and enzymes. Again, yes, receptors. Very good. And pitta is your enzymes, phosphorylation, dephosphorylation of enzymes, and then you can regulate the hormones. And kapha can be viewed as degree of gene expression. So here we can see that this is the hormones, this is my metabolic enzymes, and this is the genetic level. Viewed as the degree of gene expression as protein synthesis. This can be correlated with the ojas, that is a sense of vitality. That rules our immunity, strength, and happiness of the body or the total body defense mechanism. So this was all about what I could gather as a connection between Ayurveda and the modern medicine. Um, Basically, we can have some kind of studies conducted on free radical and its connection to the Ayurvedic medications. That is going to actually give you a final answer that what is happening at the genetic level as well as at the cellular level. Then the answers will be start and answers will start coming and the whole picture will be clear. So this is what I can give you today. Thank you so much for your attention. If you have any queries or any questions, I would like to have them and try to answer them as far as my knowledge is there. We are conducting the study of gene expression. Sorry, I just missed it. One second. Amit, can you help me in reading the...
Yes, ma'am. Yes, ma'am.
Yeah, these are some references which we have used. Can you just help me in reading the... People are sending some messages.
In chat
If we
box.
have time, we can have some...
Yes, ma'am. Yes, ma'am. And now, after session, there is a query session always,
Yeah,
as all participants know.
yeah.
Thank you, ma'am, for such a knowledgeable and interactive session. I hope that all participants get benefited from today's session. Now, the session is open for query round. Uh, Please raise your hand or write your query in the chat box. Please raise your hand if you need to ask your query. I can send you the unmute request.
And as a Santosh deemed to be university, we are open for any kind of research collaborations with any of the other institutions if you are connected to one from the Ayurveda side. It will be great for us. You can connect to me through Dr. Shubham.
Yes, ma'am, definitely. We have audience from all over the country. Approximate more than 500 participants are connected with the live session and with the live streaming on YouTube also. So please raise your hand for the query if there is any query regarding.
How to select the genes for expression of various studies? Okay. It's a very generic questions actually we you have asked. If, here there are lots of parameters which you have to first select which disease you want to study. First you have to identify the disease and then what are the gene pattern which is there in a normal person and what is the difference between the gene pattern in the disease state. Once that is identified, because you will get that in the literature. Now the question arises, what do you want to do with this defective gene? What do you want to find out? What you want to find out the polymorphism of that? You want to find out the mutation into that, or some kind of expression part. Expression part, you have to go for the protein channeling also. So there is a whole lot of protocol required, So expression part, of course, a lot of protocol is required. You have to discuss the proteins, which proteins you want to take up for the expression. Expression is always regulated by different proteins. Proteins have to be identified, and then only uh, you can make your protocol for the study. Anybody else? Any other question? Any other suggestion? Anything? I'm open to all. My mail ID is available with the Institute of Applied Statistics. You can connect to them.
Definitely. I am sharing the mail ID of our institute regarding the Ayurveda research. If there is any query regarding any help regarding your thesis and project, you want to collaborate with Santosh Dean to do university, then left the mail on the given mail ID. I am sharing the mail ID in the chat box right now. And I request it again to all participants, if there is any query, any discussion, you may send us. You can raise your hand or write your query in the chat box.
In fact, a lot of clinical trials are happening in Ayurveda medicine. The Ministry of Ayush is doing a lot of clinical trials of different drugs, Ayurvedic drugs, medicines on body processes. So that is also a very emerging field. If we don't have any more queries, I think we can close the meeting, Amit.
Yes, ma'am. Mostly, I think that all queries are answered. How can we,
Some,
Dr. Ratna Bharadwaj? Dr.
hello,
Ratna Bharadwaj, I sent you the unmute request. Please unmute yourself.
Hello,
hello,
good evening, ma'am.
good evening.
It was very informative session,
Thank you.
and I just want to ask, how can we evaluate this antioxidant effect in the body? What are the
Uh,
research protocols for that, and how can we do
First, you
in?
have to select some specific disease or some specific. In a normal person, if you want to do there, you can do it inside the cell, you can do inside the serum, blood serum. So, what exactly do you want to do? If you want to do it in a normal person and compare it with a disease state, so protocol has to be designed that way. And if you want to compare two different type of diseases as far as these antioxidants is concerned, then there is a different kind of protocol you have to prepare. There are kits available which gives you oxidant antioxidant ratio, and you can, by ELISA process, you can measure those thing, and the sample is for that is your blood serum sample. All the
Ma'am,
protocols
like
are available on different studies.
okay, like somebody is going through chemotherapy for its cancer treatment and all, na, so their immunity will be reduced.
Yes,
And we have to give them so much supplements and all things, na?
yes.
So on that basis, I want to find out
No,
and how
that is a
how can we
known
design.
case of immunity compromised immunity.
Yes,
So,
ma'am.
what exactly do you want to do there?
I want
One
to
is, yeah.
evaluate. I have some a combination of few herbal medications. So I want to assess that.
So for that you have to prepare a protocol and give your supplements, what kind of supplements you feel. First you have to identify what is missing. what kind of oxidants are being produced, what is the ratio of antioxidant and oxidants. And once you get the ratio, with the help of antioxidant enzymes, you will come to know which process is involved in there. And once that is identified, then only you can talk about the supplementation. Or you can do a pilot study. If you have some specific medicine or specific ayurvedic medicine in the mind, you can have a supplementation for 28 days or 4 to 6 weeks, which is generally supposedly is the time where this effect comes. So that way you can
The study
also
model will
compare.
be the study model we should prefer on animals or on cells.
It can be both, but depends what method you want to have and what is available with at your site of study.
Okay, ma'am.
Animal models are more comfortable because cell lines are very very costly to
Yes, ma'am.
handle. So if
Yes,
you
ma'am.
have some kind of a funding in your hand, you can go for that also.
Okay, okay, ma'am. Thank you so much.
IgG, IgM level are enough for what? IgG, IgM level will give you what?
Dr. Sarai, you're not connected with audio mode.
This is an actually incomplete question. IgG to IgM model for what? What ratio do you want to have? I could not understand. If you can just rephrase it, probably it will be easier for me to understand. What do you want to ask?
Dr. Dharmen Kumar Pandey, please unmute yourself. Dr. Dharmen Kumar Pandey.
हेलो
Yeah,
नमस्ते मैडम।
namaste.
ये बहुत ही इन्फॉर्मेटिव रहा और बहुत सारी नई चीज़ें सीखने को मिली। ये जो फ्री रेडिकल्स का जो प्रोडक्शन हम लोग के शरीर में अगर हो रहा है, तो क्या क्लिनिकली हम लोग कुछ अंदाज़ा लगा सकते हैं कि फला व्यक्ति के शरीर में फ्री रेडिकल्स ज़्यादा उत्पन्न होने से ऐसे सिम्टम्स उत्पन्न हो रहे हैं और हमने उस दवाओं का इस्तेमाल किया, तो वो सिम्टम्स कैसे डिसअपीयर हुए? क्लिनिकली कुछ हम लोग अंदाज़ा लगा सकते हैं फ्री रेडिकल्स के बारे में?
फ्री रेडिकल्स देखिए, क्लिनिकली अगर आपको अंदाजा लगाना है तो आपको ब्लड सैंपल में चेक करना पड़ेगा ऑक्सीडेंट एंटीऑक्सीडेंट आपका रेशियो क्या आ रहा है। और अगर इसमें आपके रेशियो में ऑक्सीडेंट्स ज़्यादा आ रहे हैं तो सप्लीमेंटेशंस जो अभी तक लिटरेचर अवेलेबल है वो ये कहता है कि आप नेचुरल एंटीऑक्सीडेंट्स सप्लीमेंट दे सकते हैं उनको। और सप्लीमेंट देने के बाद आप चेक कर सकते हैं कि वेदर दीज सप्लीमेंट्स आर गिविंग देम यू नो उससे उनका वो ऑक्सीडेंट एंटीऑक्सीडेंट लेवल जो है वो कम हो रहा है कि नहीं। बट इसके लिए एक प्रॉपर प्रोटोकॉल डिजाइन करना होगा आपको। यू कैन नॉट जस्ट रेंडमली डू दिस स्टडी, ओके। और ज्यादातर ये फ्री रेडिकल्स जो होते हैं वो जितने भी डिजीज फॉर्म्स हैं मोस्टली कहीं ना कहीं फ्री रेडिकल्स आर ऑलवेज इन्वॉल्व्ड। क्योंकि कोई भी डिजीज है वो इन्फ्लेमेशन करेगी। जैसे somebody is asking me, is there any relation between free radical and CRP? Yes, there is. CRP is an answer. Once your inflammation is increased, CRP levels will increase.
जी मैम। तो क्या मैम हम लोग सीआरपी को, जैसे सीआरपी के जांच तो हम लोग इन्वेस्टिगेशन करवा लेते हैं, तो इसको एक मार्कर मान सकते हैं कि इसके बढ़ने और घटने से
आप इससे ये पता लग सकता है कि इन्फ्लेमेशन है आपकी बॉडी में, लेकिन किस तरीके का। छोटी सी खांसी जुकाम जो है वो भी आपका सीआरपी लेवल बढ़ा देगी।
जी मैम।
आप कैसे आइडेंटिफाई करोगे कि किस वजह से सीआरपी बढ़ा है? उसके लिए
जी मैम।
आपको डिटेल्ड स्टडीज लगानी पड़ती हैं। सीआरपी इज ए वेरी कॉमन इंडिकेटर फॉर एनी इन्फ्लेमेटरी डिजीज।
जी मैम।
Amit, if
Thank
it
you,
is
thank you.
possible, I would like to leave the meeting.
Yeah,
कुछ
ma'am.
थोड़ा
Thank
है,
you. Yes, ma'am.
बाकी
Definitely,
आप
definitely.
सेंड कर सकते हो मुझे सबके क्वेश्चंस। अगर कुछ
Yes,
है
ma'am.
तो
Yes.
आई विल जस्ट आंसर देम।
Definitely, ma'am. The all queries are answered. We are continuously receiving the mails regarding that for positive feedback. I hope that all participants benefited from today's session. And now I would like to introduce today's resource chairperson of the session, Dr. Uma Bharadwaj, ma'am. Ma'am is currently working as a vice chancellor at Noida International University. She has also served as a vice chancellor of Modi University of Science and Technology and Maharaj Vinayak Global University. Ma'am has been honored with India's Best Citizen Award in 2019, Bharat Jyoti Award in 2018, and third CMAI CCI Technology Education Excellence Award since July 2016 and by the High Commissioners and Ambassadors of 8 countries. Dr. Umar Bharadwaj has been invited as a plenary speaker for 4th World International Conference on Wound Care 2020 Nursing and Tissue Science Amsterdam, Netherlands, March 23-24 2020. Ma'am has also invited as a speaker of 6th edition of International Conference on Plant Genomics, Horticulture and Engineering scheduled from June 8 to 9, 2020 at Frankfurt, Germany. Now I would like to invite ma'am to kindly conclude today's session. Ma'am, can you hear me?
अच्छा, ठीक।
Ma'am is not able to connect right now. Ma'am has available till 30 minutes before he engaged in a meeting, emergency meeting. Now, from the Joint State Training Center of Rajasthan, we convey our sincere thanks to all the participants for their active participation and commitment for today's session. We heartily thank the guest speaker of the session, Dr. Jyoti Batra, ma'am, who kept audience participants engaged by the way of very informative lecture and a very amazing discussion round. We also thanks for chairing today's session, Dr. Uma Bharadwaj, ma'am, and say, now this is the end of today's session. Again, thanks to all of you. Tomorrow we again, our session is on basic clinical pathology and microbiology, presented by Dr. Nidhi Pandey, ma'am. Thank you again. Thank you, ma'am. Thank you very much for your presence.
अरविंद जी,